Um, so thank you guys so much for having me. I'm so happy to be here and being able to share all this information that I've been uh, working on. Hold on a second, I'm trying to get this. Uh, okay. So excited to share with y'all. So again, my name is Andrea, and you can find me on Instagram at Florelated. Like Angel is saying, I've been a member of Central Texas Mycology for about 3 years now. Um, I I am from Austin. I grew up in exploring the Texas Hill Country, swimming in creeks and rivers. I went to Texas State University, where I got a BS in biology and an MS in wildlife ecology. And I joined Central Texas Mycological Society because I was excited to be a part of the myco community and for the opportunity to learn and to educate. I've always been really surprised at how little attention kingdom fungi gets in standard biology and ecology classes, especially just considering how integral it is to all life on Earth. I've also just been very interested in mushrooms in general, both from a culinary and spiritual perspective. I was introduced to Terence McKenna's Food of the Gods as an undergraduate and was instantly hooked on ethnobotany and the idea that human consciousness could be related to mushrooms. Um, that kind of inspired that. And I've always loved lichens. Just always, growing up in the Texas Hill Country and walking around, especially in the winter when all the leaves are off the trees, I was always just very curious about them. So I was excited to do this kind of deep dive into lichens. So today, I'm going to be talking about lichens and science, their natural history, general ecology, human culture and use, some classification and ID, and just lichens in Texas and beyond. So entitled this presentation, Lichens Are for Lovers, A Natural History of Ecological Romance. But what is meant by ecological romance? Well, we all know the importance of love in our lives and in our human experience. We witness love in the animal kingdom between the pair bonding of mated birds and the care of a mother elephant and her young. We understand the care a mother tree gives to her young through the mycelium connections in the wood wide web. But what about interspecies connections, love between species? So pet owners, including this one speaking to you now, have witnessed this between their pets, such as this love affair between my dog Duke and his fur baby Scout. So love, from all appearances, appears to be a fundamental force of nature. And in fact, I would say that it is love or cooperation, which is one of love's manifestations, and not competition, that is the driving force of evolution. So, while we'll never truly know the reasons why the first fungal and algae partnered to become a lichen, we do know from the fossil record that their ecological romance began somewhere between 400 and 600 million years ago. The early lichen fossil record isn't very clear. It can be very difficult to tell lichen fossils apart from other fossils. But evidence leans towards lichens being younger than the most complex plant fossils. So the fossil image up here, let's see if I can get my little pointer up. This one here, this is from a study published in Nature in December of 2012. And this study suggests that ediacaran fossils, which are the fossils that are pictured here in this image, were not marine animals, but were actually terrestrial lichens. If true, this would mean that the earliest large life forms may have appeared on land long before the oceans were filled with creatures. These fossils are dated to 530 million years ago. The bright orange fossils here in the bottom, these are currently what are believed to be the oldest lichen fossils ever found at 600 million years old. And these were found in South China and the Doshantuo Formation. And this study suggests that some lichen evolved in the seas, as this is an oceanic formation. And then finally, the leafy looking fossil on the right is what was previously considered to be the oldest confirmed lichen fossil ever discovered. and it is from the early Devonian, about 400 million years ago, from the Rhynie Shirt near Aberdeen, Scotland. And at about this same time, there were several unusual plant-like organisms living on land, including the mat-like Nemophostites, the crust-like Spongiophytes, and the huge log-like Prototaxis. Under the microscope, these can be seen to be made up of intertwining filaments, and some paleontologists have proposed that these fossils are unusual lichens. Spongiophytes might plausibly be lichens, but the microscopic filaments that make up nemophotites and prototaxis lack the typical branching patterns of fungal hyphae and probably represent an isolated offshoot of algal lineage, unsuccessful experiments, as it were, in colonizing the land, which left no descendants. So the study and classification of lichens began in 300 BCE when the Greek naturalist Theophrastus introduced the term lichen to refer to patches on olive trees that he assumed affected their growth. We're not exactly sure today if those actually were lichens that he was describing, but the term has stuck. The Persian medic and natural historian Ibn Sina wrote about lichens in food and medicine in his book Al-Qanan fi al-Tib, which translates to the Canon of Medicine. And then in 1686, Englishman John Ray published Historia Plantarum, which included accounts of known lichen species. In 1699, Robert Morrison's book Plantarum featured the first series of more than 100 fully illustrated lichen species. However, he did not refer to them as such, instead calling them musco fungi, or moss fungi. Then in the early 18th century, Pier Antoni Micheli and Johann Jacob de Lennis produced detailed classifications of nearly 300 species. The universal use of the term lichen, began in 1753 with Carl Linnaeus's introduction of scientific nomenclature in his work Species Plantarum. Erik Icarius is known as the father of lichenology. He was a Swedish botanist who pioneered the taxonomy of lichens and was famously the last pupil of Carl Linnaeus. And during his lifetime, he classified over 3,300 species of lichen, separated into 40 different genera. And over his lifetime, Icarius collected over 5,500 specimens of lichen, most of which today are housed in the Botanical Museum of the Finnish Museum of Natural History. And I've just kind of included a few words that are used to describe lichens. My favorite is endolith, and these are lichens that grow on the inside of rocks. I think that's so interesting. So up until this time, scientists assumed that lichens were some sort of plants. Again, the term moss fungi, it was assumed they were related in some way to plants, just like moss was. And it wasn't until 1867 that a Swiss botanist by the name of Simon Schwendener used technology in the form of improved light microscopy to see that lichens were actually made of what appeared to be filamentous fungal structures and green cells, which he assumed to be free-living algae. At the time, his claim was considered to be very controversial and was rejected by many of the leading lichenologists. In fact, William Nylander, a pioneering Finnish botanist and lichenologist, called it a Stultitia Schwendeneriana, which translates to Schwendener the simpleton. So this is very common in a lot of science where someone comes up with new information, it's considered very controversial, and they're kind of ridiculed for their assumptions. But despite the protests from his peers, Schwendener did not back down, and in fact, other researchers were able to confirm his results and interpretations. First, other botanists re-examined the lichens under the improved microscopes, and they found as well that the green cells inside lichens were identical in structure to free-living algae. The fungal components formed hyphae surrounding the algal cells, and fungal reproductive structures were observed, such as asciocarps and spores, clearly showing that these were Ascomycete fungi. So a key observation was that when the lichen fungal spores germinated, they did not form a lichen unless the algal cells were present. And that strongly suggested that the fungus could not make a lichen on its own. So next, researchers such as Anton de Berry, Albert Bernard Frank, and Hermann Hellriegel demonstrated that the fungal and algal partners could be grown separately in sterile culture. And when done like this, each behaved like a typical fungus or alga when alone. So this directly contradicted the older idea that lichens were single organisms. And then the strongest confirmation came from resynthesis experiments that were conducted in the late 19th and early 20th century. So several scientists worked to lay the groundwork for this, starting with Albert Bernard Frank in the 1870s and 80s. And the idea was just to grow fungal spores and algal cells in isolation, and then to bring them together under controlled conditions. It took many iterations, but eventually scientists were successful in creating artificial reconstitution. And this demonstrated that the lichen body is constructed through partnership and not inherited as a single organism. The fungus actively incorporated the algal cells into its structure. So George Barr is often credited with one of the clearest early demonstrations of artificial reconstitution. He isolated fungal spores from a lichen and cultured them with an algal partner separately, and then recombined them under sterile laboratory conditions and observed the formation of lichen-like structures. And then definitive proof was attributed to Eugene A. Thomas in the early 20th century. Again, he germinated fungal spores, introduced compatible algal cells, and produced structured, stable lichen thalli. And at this point, the scientific community broadly accepted the dual nature of lichens. So what is a lichen? A lichen is a complex life form that is a symbiotic partnership of two separate organisms, a fungus and an algae. The dominant partner is the fungus, which gives the lichen the majority of its characteristics, from its thallus shape to its fruiting bodies. And then the alga can be either a blue-green alga or a blue-green, or a green alga or a blue-green alga, which is otherwise known as a cyanobacteria. And many lichens will actually have both types of alga. And the fungal component is known as the mycobionts, and the photosynthetic algae partner is called the photobiont. The fungal partner forms a matrix or a network that provides structure and support for the lichen, while the algal partner performs photosynthesis, producing sugars that the fungus uses. So this matrix, composed of fungal hyphae, houses the algal cells, creating the symbiotic relationship where both organisms benefit. The lichens are foundational to the endosymbiotic theory, representing a classic example of two different organisms living together to form a new organism that can survive harsh conditions where neither could alone. And this concept of different life forms merging inspired the later theory that mitochondria and chloroplasts originated from engulfed prokaryotes, forming the basis for complex eukaryotic cells, a process known as symbiogenesis. So lichens serve as a tangible, complex model of such interactions. And this theory was actually first proposed in 1905 by Russian scientist Seymour Skwasky. And his 1910 paper fleshed out this initial expose for what we call today endosymbiotic theory. And then in the 1960s, biologist Lynn Margulis put forth the notion that the foundation of evolution is two organisms of different species benefiting through coming together and synchronously reproducing. And she contended that evolution had a jump start with the help of unicellular organisms joining to form hybrids over generations, leading to multicellular organisms with more complex DNA. And Margulis supported this theory through showing mitochondria have their own nuclei with different genetic makeup than their DNA. Her theory at the time subverted traditional theories of evolution, suggesting that evolution has its roots in symbiosis rather than mutations occurring within a single organism's genome. And although her theory would prove to be critical to studying evolution, it was considered very controversial and even ridiculed at the time. And the manuscripts containing her theory were rejected 15 times before finally being published in 1967. But her revised version of her thesis, titled Symbiosis and Cell Evolution, while still controversial when published, is today widely acknowledged as evolutionary doctrine. So what you might be asking right now is a question that I had is, if lichens do not have a unique genetic inheritance, how is lichen morphology passed down? Well, lichens can reproduce in two ways. One is by dispersion of fragments of the thallus. So these fragments contain both fungal and algal cells, so the morphology is passed down directly. The genetic combination is preserved and the structural reorganization reestablishes itself as it grows. The other way that lichens reproduce is through the fungal partner only. In this way, the fungal spores disperse alone and must encounter a compatible algal partner in order to reorganize into a lichen. So here's the key. The fungal genome contains the developmental program for thallus construction, but that program is activated only when the fungus detects specific biochemical signals from a compatible photobiont. So if this fungus associates with a different algal strain, then the resulting thallus may look slightly different or grow at a different rate or exhibit some sort of altered chemistry. But the core architecture still reflects the fungal species. The thallus morphology is not stored as a blueprint of shape in isolation. Instead, what's inherited is the fungal genes controlling hyphal alga organization, the genes regulating symbiosis recognition, signaling pathways responding to algal metabolites, and cell adhesion and tissue layer formation mechanisms. In other words, the morphology is an emergent property of genetically encoded interaction rules. This has been described as being similar to like ant colonies, which have a structure without a colony blueprint, and multicellular embryos self-organizing from genetic signaling gradients. So there are about 20,000 known lichen species, but what is meant by a species is different from what is meant by a biological species in plants or animals, or fungi, where beings the same species implies that there is a common ancestral lineage. Because lichens are combinations of two or more different, or even three different biological kingdoms, these components must have a different ancestral lineage from each other. So by convention, lichens are still called a species and are classified by their fungal partner, not the algae or cyanobacteria that they are associated with. So lichens are given the same scientific name as the fungus, which may cause some confusion. But alga will bear its own scientific name, which has no relationship to the name of the lichen or fungus. So some general characteristics of lichens. They are among the hardiest and most resistant of all plant organisms. They can withstand long periods of drought, incredible heat, and freezing temperatures. The fungal thallus can anchor to a rock, a tree, or the ground, while the alga manufactures the food for the partnership from the sun's energy. There's sometimes a misconception that lichen damage the plants that they grow on, but this is not true. In their lichen partnership, fungi do not act as decomposers. Rather, they simply harvest the energy created by the alga from the air. So lichen are poikilohydric organisms. Poikilohydry is the lack of ability, structural or functional mechanism, to maintain and or regulate water content to achieve homeostasis of cells and tissue connected with quick equilibrium of cell tissue water content to that of the environment. So in order to survive recurrent desiccation events, poikilohydric plants have developed key mechanisms that prevent damage to internal structures. Sucrose, a disaccharide sugar, is found in high concentrations within desiccation-tolerant plants. Sucrose is used within cells in order to help maintain the phospholipid bilayer, which stabilizes the cell structure and reduces the likelihood of damage during the dehydrating, rehydrating, and reactivation process. Lichens have exceptionally long lives, with some individuals living for several thousand years. Some species, like the map lichens shown in this photo in the Arctic, have been estimated to be over 8,000 years old. Lichens often grow very slowly, with some species adding only a few millimeters per year. This slow growth, combined with their ability to go dormant during harsh conditions and quickly revive when conditions improve, contributes to their longevity. Rhizocarpon geographicum, which is the map lichen, is a species of lichen which grows on rocks in mountainous areas of low air pollution. Each lichen is a flat patch bordered by a black line of fungal hyphae, and these patches grow adjacent to each other, leading to the appearance of a map or patchwork field. And one species of Rhizocarpon geographicum on East Baffin Islands has been estimated to be 9,500 years old. And thalli of a colony in the Central Brooks Range of northern Alaska has been given a maximum possible age of 10,000 to 11,500 years old. So lichens serve a critical role in the formation and maintenance of ecosystems. Through their slow and steady growth, they can create cracks in rocks that allow plants to take root. Lichens fix atmospheric nitrogen, enriching nutrient-poor soils. In arid regions, they form biological soil crusts that prevent erosion. And they absorb moisture from the air, contributing to the humidity levels in forest ecosystems. And they are vital food for caribou, reindeer, deer, and rodents, especially in winter. They also provide nesting materials for birds and squirrels and camouflage for insects. Lichens are what is known as a pioneer species, a species that builds a simple initial biological community that gradually gives way to other species. Over time, new plants, animals, and other organisms arrive and outcompete the pioneer species for resources, and the structure of the biological community changes. Rock-growing lichens play an important role in nature's plan by converting rocks into soil. They produce a weak acid, which slowly dissolves the minerals, forming tiny cracks in the surface of the rock. And as the tiny cracks appear, the threads of the fungus dig deeper into the rock. And these threads shrink in dry weather and then swell in wet weather, enlarging the cracks. And then during cold weather, the moisture in these cracks freezes, often breaking or splitting off pieces in the rock. This disintegrating process does not happen overnight. And in fact, little progress is noticeable even after a century. But as time passes, the lichens slowly etch their way deeper into the rock, forming a hollow. Dead lichens and rock particles slowly accumulate in this hollow until there is enough soil to support mosses. More time passes, the mosses grow and die, add themselves to the soil, and then gives way to ferns, flowers, grasses, until thousands of years later, there might be even trees. So these pictures that I took on a recent trip to Enchanted Rock State Park, one of my favorite state parks, kind of shows this process. You can see these lichens growing on the granite here. And then here is where over time we've seen that there's been some erosion, water piling up down here, maybe some soil. And then here's more of a fully developed stage in the succession stage, more of a climax. I guess this would be the climax community in this succession. So a little bit more about lichens and animals. Many animals rely on lichen, especially like we said, caribou and reindeer, which consume vast amounts in winter, along with musk oxen, moose, mountain goats, deer, and voles. Invertebrates like moths, slugs, mites, and springtails, plus birds, use it for nesting. Here's a cute little picture of a hummingbird in a nest. Uh, Invertebrates like moths, slugs, mites, and springtails will also use it for habitat. Um, so the ruby-throated songbird uses it for a nest, but also blue-gray gnatcatchers. They use the lichen for nest camouflage, sticking pieces to the outside with spider silk to blend into tree branches, while chickadees will use lichen as a base material for their nests, making them soft and well insulated. Other species, such as warblers, northern parula, will also incorporate lichens into their nest. And then this picture in like the lower left-hand corner is a really cool insect called the lichen katydid. It's found in Central and South America, where Usnea lichen, aka old man's beard, is common. They use extreme spiny and patterned mimicry to avoid being seen by birds and other predators. And then when they're threatened, they often remain perfectly still or play dead to mimic the lichen. Oh, and then this is a picture of the reindeer lichen here at the bottom too. A good picture of that. So lichens, the ability of lichens to indicate air quality has been recognized since the 1860s, when botanists in Paris discovered that lichens thrive in areas where the air is clean and suffer in areas where it's dirty. Early investigators focused on the direct effects of sulfur dioxide stemming from burning coal. And then over time, scientists discovered that lichens were also good indicators of the regional effects of acid rain caused by longer range transport of sulfur dioxide and other industrial emissions. Lichens are also sensitive to ammonia and nitrates that drift from agricultural areas where fertilizer is used, and they accumulate heavy metals such as mercury from power plants and lead and zinc from mining or smelters. So the U.S. Forest Service has been collecting lichen data since the 1970s with nationwide emphasis beginning in 1993. In 1993, the NFS and the Forest Inventory and Analysis Program have collected lichen data on forest land, and then they've created a database uniting this data with partnering agencies and researchers, including data on both community composition and in elemental analysis of lichen tissue. And as of 2017, the database housed 171,000 lichen records and 15,000 lichen tissue samples collected on over 10,000 plots. And it's usually uploaded and updated annually to include new data and add new functionality. Most of the data that they collect focuses on macrolichen, the non-crustose epiphytes in forest habitats, although some data on ground, rock, and crustose lichens is available. So ethnolichenology is the study of the relationship between lichens and people. So lichens have been a traditional food source around the world, particularly in parts of Asia, North America, and Scandinavia. While nutrient dense in carbohydrates, they generally require thorough cleaning and boiling, often with ash, to remove any mildly toxic acids. And common edible species include rock tripe, Iceland moss, and reindeer lichen, which are used in soups, bread, salads, or as thickeners. In this photo in the upper right, it shows two freshly cooked loaves of willa, Bryoria frementi, which is an edible dark brown horsehair lichen that grows hanging from trees in Western North America and Northern Europe and Asia, and is an important traditional food for some First Nations in North America. Lichen also has long been used as a dye. It was actually first mentioned in the Bible, Ezekiel 27:7. It was apparently first used for red and purple dyes by the people of Tyre thousands of years ago, and the desired orchidal purple colors were derived from Rochella tinctoria and other rock lichens of the same genus. The use of lichens in ancient Egypt for embalming dates to the 18th dynasty. Rasudavernia furfuracea, known as tree moss, was found in an Egyptian vase. So in the embalming process, once the organs were removed, the body cavity was packed with lichen, sawdust, perfumes, and spices. And the tree moss was employed due to its preservative and aromatic qualities. Also, it's highly absorbent and contains antibiotic substances, and this helped inhibit the bacterial decay of the mummies. And then another discovery is that the Egyptians would grind and mix the tree moss with their flour for bread. And then the bread was placed with the mummy and thought to be the first meal for the mummy in its afterlife. The tree moss was imported in shiploads from the Grecian archipelago to Alexandria. And today, embalming fluids are colored from the lichen dye or chill into a product called cudbear, illustrating how a historical procedure can influence future practices. And lichens have a long history of use as medicines and are actually still being utilized today. There's lots of studies into how they can be used. They have a lot of anti-cancer properties, for example. But one interesting example that I chose is this one here in the lower left-hand corner. And this is a more recently discovered lichen called Dictyonema chirarni. I know I mispronounced that, but it is a recently discovered species of lichen from the Amazon. It has a cultural history of use amongst the Warani, a native Amerindian tribe living in the Amazon rainforest. And it was reported to have psychedelic properties and produce psilocybin. Though due to the rarity of the specimens, this has not been confirmed. I just thought that was interesting that there was a lichen that was producing psilocybin. So lichens can also provide a biological century scale diary for estimating gravestones or other stone monuments through a process called lichenometry. So as they grow in slow, consistent, and measurable circular patterns on the stone, people can measure the diameter of the largest lichen, thallus, and calculate how long a surface has been exposed, as larger colonies indicate older, undisturbed, or historic stones. Lichens generally grow by fractions of a millimeter each year, roughly 0.0685 centimeters a year. So to determine the age, the largest circular lichen patch on a gravestone is measured, and then it's compared to a growth curve that's established from a nearby dated gravestone. We know that it usually takes about a decade for the lichen colonization to begin after a stone is placed, and then as time passes, new species may appear with younger, faster growing lichens, sometimes cutting through older ones, indicating a succession of colonization. Lichenometry is especially useful for dating unmarked or weathered graves, and they can confirm the grave sites if the grave site predates known historical records or ownership. And I actually took these photos of lichens on some gravestones when I was in East Texas visiting a historic graveyard in November. And so many of the stones had actually been completely covered with lichen, and you couldn't even read them, but they looked really cool. So when we characterize or identify lichens, we're usually grouping them by the shape of their thallus. And there are three basic growth forms. There's the crustose, foliose, and fruticose. Crustose lichens are crust-like and adhere tightly to the surface, while foliose lichens are leaf-like and not tightly bound. Fruticose lichens are shrubby or tufted with three-dimensional structures. So here's some example of some crustose lichens. They form a crust-like layer on the surface they grow on. They can grow on rocks or tree bark, and they can be very difficult to remove without damaging the surface. Here's some examples of some foliose lichens. They have leaf-like lobes that are not tightly bound to the surface, and they can be more loosely attached to rocks, wood, or other surfaces. And then finally, the fruticose lichens. And these are a type of lichen that are characterized by their shrubby or coral-like three-dimensional shape or growth structure, often resembling tiny trees or bushes. And they can grow upright or hang down and have branches that are typically round in cross-section. So we can identify lichens based on their thallus shape. Another important mode for identification is the secondary metabolites. So lichens produce over 1,000 unique, mostly fungal-derived secondary metabolites, lichen acids or pigments, that are essential for survival rather than primary growth. And these compounds are often deposited on the surface of hyphae. They provide protection against environmental stress, such as UV or temperature, predation by herbivores, and they can have different properties such as being antibacterial, antiviral, antioxidant, and anti-carcinogenous. And secondary metabolites can be used for IDing lichens in the field or lab. Spot tests, chemical color reactions, quick field-based tests using reagents like potassium, hydroxide, sodium hypochlorite, and paraphenylidamine to detect compounds based on color changes in the medulla or the cortex. Thin layer chromatography, TLC, is the standard method for separating and identifying lichen substances by comparing their values and colors under UV light with known databases. Analytical techniques like high-performance liquid chromatography, HPLC, and mass spectrometry are used for precise identification and quantification. And then software resource tools like metabolites or LIAS metabolites are used to identify compounds based on TLC data. So many of you might be familiar with our UV night walks, and some of you might have been on our UV night walks. And one of the things that we love to check out when we're on those walks is how lichens look under the UV light, as they always glow really spectacularly. And this unique ability is due to the specialized secondary metabolites, specifically xanthones, depsides, and fulvinic acid derivatives, which act as sunscreens to protect the organism from UV radiation. And these compounds absorb UV light and re-emit it as visible light, creating colors like yellow, blue, and orange. Another interesting compound that fungus produces, the fungal component produces, is melanin. We've been hearing a lot about fungal melanin in the news in reference to Chernobyl and the fungus that's been known to thrive in that radioactive environment. But this is because the melanin is not susceptible to radiation. And in fact, it grows towards the radiation source. So the fungus can use the radiation for food. Melanin functions as an energy transduction molecule, radiosynthesis, allowing the fungus to convert radiation into useful energy, and the implications of new energy source of fungi as autotrophs. So the melanized fungus can harvest radiation and store it as heat, and this can be beneficial in cold environments. So an example in this photo, these are the Black Mountains of Antarctica, and the pigment comes from microbes that enhance melting. And these microbes are a lichen where the fungus is producing melanin, allowing for the harvesting of solar radiation. And when glaciers begin to melt, the melanin producing lichen will fill in the holes, produce more heat, and then lead to an increase in melt and destabilization of the ice sheet. So now, moving on to our Texas Hill Country. Like we were saying, lichens are very common in the Hill Country, especially in this time of year when all of the leaves are off the tree and we're getting, hopefully, more moisture. We see them abundantly springing up on all the tree branches. Um, so this one in the middle, these are all photos that I've taken. I love to take photos of lichens, but this is one of my favorite scenes. It's just coming up on these branches and just having them be covered in lichens. I just think it's so beautiful. I love seeing all of them growing together, and then of course all the pops of color. So here's a few, going into just a few of the types that we see commonly. These are all ones that I've encountered. This is the black sheet lichen. It's a pretty common foliose lichen that we see here in the hill country. It's characterized by its large, pale green to grayish green thalli with a reticulate vein pattern on the lower surface. And then you'll find it growing on ash juniper and oak trees in open woodlands and along forest edges. This is the Texas shield lichen, another foliose lichen commonly found on tree bark, especially oak and pine and juniper. And it is characterized by tightly adnate lobe-like structures and a medulla that is K and UV plus blue-white containing divaleric acid. And it is often used for biomonitoring. Due to its sensitivity and presence in polluted areas, it has been used to monitor radionucleotides like Ra226 and Pb210. One of my personal favorites, the slender orange bush, a beautiful bright orange pop of color. Its scientific name, Teleoschistes axilis, and it's a species of corticolous or bark-dwelling fruticose lichen in the family Teleoschinaceae, and it is found in the Americas. The orange pigment in this lichen is primarily composed of peritin, and this pigment acts as a chemical defense mechanism. So when present in high concentrations, it indicates that the lichen is enduring and often thriving under higher than average levels of environmental stress, specifically high light intensity, and often elevated nitrogen or moderate pollution. Research indicates that the pigments in teleoschistes can help the lichen tolerate and protect itself from high nutrient or, in some cases, high metal loads, allowing it to survive in areas where other species might fail. And it's just so pretty. The eastern speckled shield lichen, Punctelia boleana, is a species of foliose lichen in the family Parliamiaceae. It is found in North America with a distribution extending from the Canadian province of Ontario south to central and northeastern United States and Mexico, and it grows on the bark of both deciduous trees and coniferous trees. The combination of characteristics that distinguishes this species from others are the absence of the vegetative tropicals, Isidia and Soralia, and a pale brown lower thallus surface, and the presence of secondary chemicals, proteolystrosinic acid in the medulla. So quipu lichen, Ramalina peruviana. The term quipu originates from the Andean Quechua word quipu, meaning knot. And this name is a direct reference to the ancient Inca recording device made of knotted strings. And this lichen structure, with its narrow, tangled, and branching thallus, bears a resemblance to the tangled, knotted, and braided fiber-based quipu devices. It is a fruticose lichen found all over the world, having been documented in diverse habitats such as mangroves in Australia and on certain tree species in Taiwan and China, where it thrives at higher elevations. And this specific one that I took a photo of was here in Central Texas. The rough speckled shield lichen, also known as speckleback lichen, is a North American species of foliose lichen in the family Parmeliaceae. And this species can be readily identified by the light color of the thallus underside and the relatively large lobes at the edges of the thallus, and the tiny white pores present on the top of the thallus that are characteristic of the genus Punctelia. This lichen is quite abundant and widespread in North America and usually grows on bark and less commonly on rocks. Um, Although Punctelia reducta was previously thought to have a much more expansive global distribution, phylogenetic analysis revealed that a species complex that has subsequently been split into 4 distinct cryptic lineages with more restricted distributional ranges. It is also moderately sensitive to air pollution and has been assessed as a suitable candidate for biomonitoring air quality. This one is really interesting. I saw this one growing on a rock and I didn't know what it was. And I took a lot of close-up photos of it, and it did indeed turn out to be a lichen. It's a species of diplotoma, and it's a genus of Prestose lichen. It consists of, the genus has 29 different species. This one was thriving on some limestone in the hill country. The black specks are a specific type of fruiting body called the Lecidine apothecia. And the apothecia are typically disc-shaped with the central part of the disc containing the fungal hymenium where the spores are produced. They had to get really close up to see that, but there they are. And finally, I just wanted to touch on a different type of fungi. So there are some lichen that a lichenous fungus is a member of a specialized group of fungi that live exclusively on lichens as their host. So lichens themselves can be hosts for other organisms. So this fungi, comprising over 2,000 known species across 280 genera, exhibit a wide range of ecological strategies, including parasitism, communalism, and mutualism, and they can be found in diverse environments worldwide, from tropical to polar regions, and play important roles in lichen ecology and biodiversity. Lichenicolous fungi are classified into several taxonomic groups, with the majority belonging to the Ascomycota and a smaller portion to Basiomycota. And their interactions with host lichens range from mild parasitism to severe pathogenicity, and sometimes causing significant damage to lichen communities. And that's all I have for y'all today. If you wanted to meet lichens in the wild, just a reminder, we are having that walk at Purgatory Creek next month in San Marcos, and I'll be leading alongside Gina Ruiz from Forest Bathing and Wellness. I think Gina is here tonight. Gina is a wealth of knowledge, blending ecology, art, and mindfulness to help people slow down and reconnect with the land. And she's also coming out with a book in the near future, Lichen Life. So we'll be excited to hear more about that.
Wow, that was
So
so great.
that's it.
Fascinating.
Thank you.
Thank you, Andrea. Do we have any? I saw a few comments and questions come up along the way. Does anybody want to unmute and ask their question or raise their hand?
Look at these.
Let's see.
I see people finding lichens.
Yeah, and then there's someone asked, what's the common name for Telo? I'm going to ruin this Latin, this dead
Which one?
language.
I know there's so much Latin in
Telo,
science.
Teloscistes exilis.
Was that the
A slender orange bush lichen?
was that the slender orbish? Yeah, I think that
Yeah,
was
it
that
looks like
one.
someone answered the question. So Talat asks, often several lichens share proximity. Is there any understanding of how the different species interrelate with each other, kind of like detecting detecting other lichens around them? Because I know there's some studies with
Yeah.
fungi.
Well, I think since lichens are basically just utilizing the growth medium as a structure, they're not actually they're not in competition, with each other. So I think like I often find multiple different lichen species on a branch and they all just seem to be living harmoniously. There doesn't seem to be any like you know, competition per se. So I don't know that they're like using,
Hmm. Spatial.
resources. But
Yeah,
I don't know that they're like interacting in any
yeah.
way.
Because I know there's a few species that have been looked at from, I think, microscopically, where they, or maybe even chemically, where they have like I think it's opsins. It's like the same thing that's in eyeballs, and they can use light to determine where something else is.
Hmm.
It's the same with like some trees that have like crown shyness. Like they can sort of
So
detect
they
other,
know which way to grow. They can kind of
yeah,
know which way to grow.
to prevent like
Yeah.
too much crowding or like, because
Yeah, just
sometimes when things get crowded out, like pathogens, if a pathogen comes
right.
in, yeah.
Well, and they are needing the sun's light, so they are photosynthesizing. Sorry, my cat
Hmm.
is very.
Yeah. So Holly asks, can any species of fungi become a lichen?
So I kind of dug into this because I was really curious, like what happens, like when the ones are grown separately, like when they grow together, they form the thallus and the morphology of the lichen. But when the fungi grows by itself, it's just sort of just the hyphae. It doesn't have like a specific body structure. And the fungi that has formed relationships with lichen has been lichenized. It's called like lichenized fungi. So it's like had this relationship for a long time. So I don't know if there's been any like discovery of like new fungi becoming lichenized in historical times. Um, I don't know. But I know that the fungi that is a part of the lichen relationship is known as lichenized fungi typically.
Hmm.
And I guess it holds that memory of how to like create the thallus. So this is actually like a really I kind of dived deep into this because I was really trying to wrap my mind around around it because I was like, how does it know to form the lichen? How does it remember if it doesn't have that DNA? Because we think like when the DNA holds the blueprint, for for the species when the species reproduce, but without that, how does it know? So that was
What's in the ether telling it what to
yeah.
do?
So that was kind of like the fungi has in it, those memories, those genes that know like, okay, I can recognize this algae, and
Mhm.
then when it is with that algae, then that produces like, okay, now it triggers, these other genes to come on that turn on, and suddenly they're like, okay, we'll grow into a thallus.
Yeah. I remember Gina saying on the walk too, they're starting to find that there's like other like yeast or more like bacterial relationships too, because we're learning like that there is like some, sensing and like bacteria, there is communication happening within between bacteria or single
hmm
cell organisms too.
Right. Right. Yeah.
And so it's not like
They're
everything's out there on alone. We're
yeah.
interspecies connected.
They're connected.
Should we do
It's
the book giveaway and then we could do some
oh
more
yeah.
questions?
Yeah.
Okay, so I did a random number and then it picked the name Jen Mudge. So Jen, I just need your mailing address. So if you can mail that, email me your mailing address. The email is Central Texas Mycological or Central Texas Mycology info at Central Texas Mycology. So if you can just send me your address, I will get that out to you probably tonight.
That's so exciting. Thank you.
It's a great
Thanks for
book. You're gonna
coming,
love it.
Jen.
Is it like Texas spelled out or just TX in your
Oh, TE, Texas spelled out,
okay.
centraltexasmycology.org.
.org. Okay.
Yeah, and I
Great. Thank
dropped
you
it
so
in.
much.
Yeah. Okay, so back to questions. Let's see.
So somebody asked if lichens have a mycorrhizal relationship, and they do not. So they do not have any sort of interaction with the organism. If they're growing on the tree, they're just attached to the tree, but they do not take any nutrients from the tree. They don't decompose the tree. The hyphae do not act like hyphae. Like in a normal fungus, the hyphae would be pulling nutrients, from the tree or whatever, but not lichens. They do not do that. Does not act like a fungi. The fungi does not act like a fungi when it's a lichen. It acts like a plant.
Yeah, and this is more of a general question for Central Texas mycology. Do we have a database of academic resources? We have not subscribed to any of these gate kept academic publications. But if there's like a paper you're looking for, if you see something that is behind a paywall and you want it, we usually know somebody in the academic spaces that can get us the paper. So that's kind of just how we currently operate. I know like some mycological societies like will hold all of the things, but really we just kind of, whenever there's a paper that I want to read and it's behind a wall, I just find someone I know that's has a expensive subscription.
There's usually somebody in grad school.
Yeah. So yeah, if there's ever something, we can always help you get to it if you don't know anybody that can help you get past the wall, the 12-foot wall.
I was going to respond. Stephanie asked if there was a point in the fungus's life that they stop responding to the presence of algae, or that they can grow a thallus for the algae to attach to. And so during the course of their interaction as a lichen, they don't ever separate. So they're just interacting as a single organism. The only time that they would stop interacting is if they were like forcefully removed from each other, which is like during one of the experiments, where they would separate the fungi from the algae. And then in that case, when the fungus is separated from the algae, it does not grow a thallus. It's just sort of undifferentiated hyphae. It doesn't have like a shape.
I don't know. Got your address, Jen. Let's see. Oh yeah, Rachel, that was a good suggestion to add on what I was saying. Emailing the authors always works too. Yay.
Yeah. Well, y'all, I saw some of y'all are ordering the book. I highly do recommend it. The photos are really great. It has some really cool information in there. Some of it I used for this presentation. Some information was from other resources, but this one was definitely worth having. And always
Yay.
like good photos.
Yes. Great. Well, I'm going to go ahead and end the live stream
Okay.
since we don't have any other questions.
Thank you, everybody.
Thanks for
letting
coming
me talk.
tonight. Yeah, and Andrea, if you want to stick on, I can talk to you about that email I sent to you today.
Okay.